Okay, so first I need to say sorry everybody. It seems that I do not know how to use the forum function on Madoc, all right? So some people have told me they were waiting for Microbe of the Week and nothing arrived, and I was sitting there waiting for answers all evening and nothing arrived. So I sent the question out at 7 p.m. with the forum function, and I got an email with my message. I thought, oh, that's fine, this way it works, right? Cool. So then as time went on, I thought, no answers, what's happening? And then I had to have like my dinner and do something else with my evening, so I didn't resend. So what I suggest we do is I'll send the question out again, this time so that you get it this afternoon. So when would be a good time for you? That is not going to mess up your evening, for example. 4 p.m. Hey, well, you never know. So 4 p.m., all right? I'll send. Okay? So I will be checking my email from then. So if you don't get anything this time, email me and say, there's a problem, I did, okay? Okay, so that's what we'll do. Now, this morning we're going to carry on with the structures of the bacterial cell, carrying on with what's on the outside, finishing up with what's on the inside, and possibly, if we finish, we'll go on to the beginnings of bacterial identification and classification. Right, so we stopped before LPS, lipopolysaccharide, which is what makes up the external leaflet of the outer membrane of gram-negative bacteria. LPS has this structure, lipopolysaccharide, so lipid and polysaccharide. Now the lipid part is lipid A, which makes up the outer leaflet of this membrane. And directly attached to it, there's what is called a core oligosaccharide. And you certainly do not have to remember this structure. Okay, so the sugars in here are a bit unusual. These ones you recognize, glucose, galactose, okay, N-acetylglucosamine maybe. This one is very particular. KDO is keto-deoxyoxylosanate. But you don't need to know that. Some kind of unusual sugar molecule, all right? And the core oligosaccharide is highly conserved between all gram-negative bacteria, in all gram-negatives. And then as you get further up the polymer, then the next structure is a kind of a repeated motif of sugars. So when I say repeated motif, you can have like, I don't know, 3 sugars, or 1, 2, 3, 4, 5 sugars like this, and then this will be repeated dozens of times. So you have a long molecule that is up on the outside of the outer membrane. So that's the structure of this thing. What is its function? Well, generally, it's to protect gram-negative bacteria from chemical environmental insults. So for generally speaking, okay, so the outer membrane is going to be sensitive to detergents and solvents. And the hydrophilic sort of layer that is going to be formed by all of these this polysaccharide here. This will prevent detergents and solvents from getting to the membrane and disrupting it. So it's chemical protection. Also for gram-negative bacteria that live in the gut, to colonize the gut, they've got to pass through the stomach and they've got to pass through the small intestine. And they have to resist the enzymes in the chemical conditions of digestion. So LPS will protect the membrane from being disrupted by bile salts, which are normally involved in digestion of fat, right? Okay, so that's how gram-negative bacteria can not be digested. They also protect bacteria against, well, some antibiotics. For example, if you didn't have LPS, any kind of hydrophobic molecule could diffuse freely across the membrane, including some antibiotics. So why do these guys need to have a protection against antibiotics? Of course, most antibiotics are synthesized by other bacteria. They're trying to kill each other all the time. So the outer membrane, the reason why it exists, one of the theories is that it's a kind of defense mechanism that bacteria use against to protect them from other bacteria. Now, that's what it's useful for gram-negatives. When you another term for LPS is endotoxin, because if you have an infection caused by gram-negative bacteria, your body will detect the presence of LPS and start an acute reaction. So there are a number of receptors of the innate immune system that recognize this conserved structure that's at the base of LPS. So this is highly conserved, highly specific for gram-negative bacteria. So the receptors that detect lipid A and the core oligosaccharide, they're telling your body, you've got a gram-negative bacterial infection. So this is what's going to start up inflammation. Now that's, and then so detecting endotoxin will boost up your immune system, start off a fever, start off local symptoms like diarrhea if you've got a gut infection. And all of this is a useful response for your body to get rid of the infection when it's localized. If you have a generalized gram-negative septicemia, then you can have a massive activation of your innate immune system, and this will lead to septic shock. And mostly it's because of the presence of endotoxin, that is LPS, lipopolysaccharide. And the last thing you need to know about this is that the O antigens are variable between strains of bacteria. So even within the same species, these repeated O antigens will vary. So they're useful for identification at the infraspecies level. If you want to distinguish two different strains of Escherichia coli, for example. Okay, so it's not really, yeah, that's what it says, O antigen. So now this is not really in the program for L2, but I think it's useful for you to know to understand how LPS is synthesized. And there are two different pathways for the core, the lipid A and the core oligosaccharide, and for the O antigen repeats. So the core oligosaccharide is synthesized on the cytoplasmic face of the plasma membrane on lipid A. So once this core antigen has been synthesized, it gets flipped over into the membrane, so it's now on the periplasmic face. the O antigen repeats are synthesized by another pathway. So you get your kind of oligosaccharide repeat that's synthesized again on the cytoplasmic base, connected to another lipid molecule, flipped over so that this is in the periplasm, and then one repeat is going to be transferred onto a growing chain of repeats by a transglycosylase. So you get a growing chain of O antigen repeats, and then after a while, the whole thing is going to be transferred onto the core. What do you think this could be? This lipid transporter. No guesses? Yeah, it's a fatty acid, and it flips a repeated subunit over the plasma membrane, which is then going to be transferred onto a big chain by a transglycosidase. Does this remind you of anything? If I'm asking you this question, it means that it should. Yeah. Yes, yeah. So this is synthesized on bactoprenol, the same thing as for peptidoglycan. And this idea that you transfer a repeated subunit onto a big polymer with a transglycosidase also is very similar. Okay, you've got a similar kind of process going on. Now, once you understand this, you realize that, okay, this is why the O antigens can be variable, and this can remain the same. Okay, so in different types of gram-negative bacteria, this part of the biosynthetic pathway will be identical. Okay, so you've got the same enzymes producing the same structure. But this part here, this is going to be different. Okay, so you'll have a different repeating subunit produced, and then afterwards, okay, it can be the same. So that's how LPS is synthesized. And then in the second step, you've got this whole huge, this big LPS structure. There's a big protein complex here in the outer membrane that's going to pick up the whole thing and push it through to the outer membrane. And I have absolutely no idea how this works, okay? Now, a similar kind of structure that you can find on the outside of gram-negatives and gram-positive cells is the capsule. And you can sometimes see the capsule just with light microscopy. You can see this kind of halo around the bacterial cell. That's the capsule. And by electron microscopy, you can see, here's the cell, and there's a kind of like almost like a glycocalyx around the cell here. So this is some kind of repeated polymer, can be present on gram-positives or gram-negatives. And for the first time, this is a facultative structure in bacterial cells. So peptidoglycan, LPS, these are necessary structures. They're required for the bacteria. Capsule antigens are not. They can be present or absent. And once again, they are used for identification of bacteria. They're called the K antigen. K for Capsule in German, apparently. Now, why do bacteria produce these things? Once again, it's a kind of protection against detergents, antibiotics, bacteriophages, viruses that attack bacteria. They need to bind to a receptor on the surface of the bacterium. And if you surround the whole of the surface with a capsule antigen, these viruses won't be able to bind to their receptor. So this can have a protective effect. Also, they can be useful to protect against desiccation because they're often quite hydrophilic, so they can retain water inside this layer here. And if bacteria are isolated on a surface, then it will prevent them from drying out. And in terms of pathology, capsule antigens are often very important virulence factors because they will prevent bacteria from being phagocytosed by neutrophils. So, for example, Salmonella enterica subspecies typhi. Different types of Salmonella can infect your gut, and they can cause salmonellosis, gastroenteritis, and some can cause typhoid fever. So salmonellosis is a localized infection restricted to the gut. Typhoid fever is a systemic infection, bacteria in the blood. And the difference is the strains that can cause typhoid fever have a capsule antigen. And those that remain localized don't have a capsule. So it's important for pathology in many bacterial infections. Now, once again, you don't actually have to know this, but briefly, the biosynthetic pathway is, okay, once again, you have a synthesis of, okay, yeah, so what are they made of? I forgot to say about this, okay. Polymer. Okay, capsule antigen polymers are mostly polysaccharides, sometimes polypeptides. Anthrax bacillus anthosis has a polypeptide capsule. Some bacteria have a capsule-like structure made out of a lot of copies of protein. But mostly we're talking about polysaccharides or polypeptides that are not synthesized by the ribosome. So for oligosaccharide capsules, this is the biosynthetic pathway. Once again, you've got the repeated subunit. It's going to be synthesized on bactoprenol on the cytoplasmic face of the plasma membrane, transported to the periplasmic face. Then you have transfer of the repeating subunits by a transglycosidase. And then you'll have another kind of big protein complex that's going to shift the whole thing right to the outside. So the reason why I spent time, just briefly presenting the biosynthetic pathways of LPS and the capsule antigen is just to show you how similar they are to peptidoglycan synthesis. And that's because all of these structures is the same problem for bacteria. You've got to synthesize a big polymer on the outside of the cell. It's a complicated problem. It was solved for peptidoglycan synthesis. And then these other structures, LPS, capsule antigens, they're adaptations of the same kind of mechanism. Okay, once you've got a solution to the problem, you can use it again to produce other structures. So that's how these things are synthesized. Now, other structures on the outside of the bacterial cell that perform interesting functions are fimbriae and pili. So when I talk about this, I don't mean these big long filaments here, which are flagella, but just the kind of short hair-like projections that you can just about see on the surface of this bacterium here. Now, there are two names for these structures because they were observed for and described independently by two different microbiologists. So one of them decided to call them fimbriae. So this is like a thread in Latin, and the other one threads, and the other microbiologist called them pili. So this is hairs, right, like pilus. So you can have hundreds of these per bacterium, narrow diameter, and they have a bunch of different functions. Adhesion, binding to host cells, secretion of virulence factors, and gliding motility. And as well as that, you have F. pili or conjugation pili, which are slightly larger, slightly wider diameter, fewer per cell, and they're required for conjugation. That is the transfer of DNA from one bacterium to another. So they produce these kind of big tubes, go from one bacterium to another. Now, there are quite a lot of different functions here. And fimbriae and pili do not have the same structure. There are different types. So which type can produce, can mediate which function? Now, once again, this is going beyond really the program of your L2 microbiology, but I like to present this because, you know, for a lot of things in biology, understanding the function requires you to understand the structure, okay? And the functions that different biological objects can perform really depend on, you know, how they're made up. So I'm going to go through this. Right, these different. I'm going to talk a little bit about these different types of PD, and I'm going to go from left to right on this slide. Now, originally, way back before any of this was known, okay, these are the kind of like cartoons of the molecular structures and their biosynthetic pathways. Before this was known, fimbriae and pili were classified serologically by, you know, a bunch of antibodies that cross-reacted with different kind of fimbriae. So there were type 1, type 2, type 3, type 4. And in fact, this classification has kind of been replaced by a more structure-based classification. However, type 3 and type 4, these names still exist because the original class corresponds pretty well to the different types of pili. Class 1, class 2, they don't exist anymore. Okay? Right, so first two types here, chaperone, usher, and curly pili. Their function is only adhesion. All right? How are they produced? Okay, they're anchored, and I should also say gram-negative bacteria. So we've got plasma membrane, periplasm, outer membrane. So chaperone, usher, pili. The main filament here, there's an adhesion on the tip, and then most of the length of the filament is with the filament protein. So proteins that go up to make up this filament, they're synthesized in the cytosol, secreted out to the periplasm. Then they will bind to a chaperone, which brings them over here to the base, and then they polymerize. So how these filaments are produced is that they will be extruded from the surface of the cell up to a certain length, and then they stop. CU, curly pili. So these are called CU, chaperone, usher, pili. Curly pili is kind of similar. You get the subunits produced in the cytosol. They're exported out into the periplasm. And then there's another exporter which will just put them to the outside surface of the cell. And they will go and bind onto the top of this expanding filament. So all of these structures are for gram-negative bacteria. Gram-positives have something like this. Okay, they can have a pili that are a little bit like curly pili, but they'll just stick onto the top of the peptidoglycan layer, the cell wall. So once these pili have been synthesized, they can't do anything else because the location of the polymerization is on the outer membrane or on the surface of the cell. And the bacteria can't really control anything afterwards. Okay? So they're just structures, filamentous structures on the outside of the cell. They can bind to something. That's it. Okay? So they have a function in adhesion. Type 4 pili. Now, they're synthesized in a very different way because what is happening here is you have a kind of more complicated structure that traverses both the internal and the outer membrane. And the subunits that make the filament are added onto the base plate here. And this is controlled by a complex that's in the plasma membrane. Requires the hydrolysis of ATP, and it can be reversed. Okay, so the polymerization, you can add subunits to the base, and then this filament will extrude from the cell, and you can take away subunits from the base, and then the filament will be retracted. So retraction possible? Yes. And that's why type 4 pili are used for gliding motility. What does that mean? Or what does that entail? That means movement of a bacterium over a surface. So you've got, this is your bacterium. It's going to send out type 4 pili out to the front because it's responding to some kind of chemotactic signal. It will bind here, and then the filament's going to retract. So this will pull the bacterium along. Okay, so overall, this guy can move over a surface. And that's possible because these pili can extrude and retract. Can't work for these guys. Type 3 secretion pili. So how do these work? Once again, you've got a big complex structure in the inner and the outer membrane. This time, the subunits that make up the filament are not added at the base, but they go through the filament here. It's hollow, it comes out at the top, and they're added on at the outside. What does that mean? That means that this filament is hollow. Okay, so it's empty on the inside, it's not solid, it's hollow. That's why it's got a bigger, wider diameter. And it means that it can be used for secretion. So this kind of type 3 pili are for, they're like needles that can inject bacterial proteins into a neighboring cell. So often they're associated with virulence in pathogenic gram-negative bacteria. These bacteria can have a type 3 secretion apparatus. It will inject cells in our body with some kind of horrible bacterial protein that will, I don't know, well, alter the host cell. And the last kind is a kind of, well, it's called a type 4 secretion pili. So it's type 4. Why? Because once the structure is similar to here, and the biosynthetic pathway is the same, so that the filament proteins, they're added on from the base. So the filament can extrude and be retracted. But it's hollow, like a type 3 secretion pilus. And so, well, proteins or DNA molecules can be secreted. So type 4 secretion pili, the conjugation pili, F pili, they're a specialized type of type 4 secretion pili. So these are the ones that are involved in transfer of DNA between bacteria. Okay, I think I'll show a quick video. Okay, I'll just show you one. So what you're going to see is, oh, stop. The red color on this projector just seems to not exist because the bacteria, I can see them in red, but and the filaments are in green. Okay, so you can't actually see the cells here. Okay. Oh, yeah, okay, well. That's unfortunate because up in the top here you've got the cell and you can see that it's attached to another cell over here and it's retracting the filament, the F pillars that's holding them together, and they're going to, and the two cells are brought together. Okay. So I can see it on my screen, but okay. Right, so last structure on the outside of the cell, on the outside of the bacterium, are flagella. And the position of the flagellum on the cell is, well, it's described by these terms. So monotricious, just got one flagellum. Lophotricious, several flagella at one pole, one end of the bacterium. Peritricious, you've got flagella all over the place. Pseudomonas has just got one. Proteus is going to have the flagella all over the place. Now the flagellar rotor is like this. So you have a base plate structure which is anchored in the inner and the outer membranes, a hook structure, and then the filament. And this rotor is going to turn round using proton motive force. So protons that are in the periplasm will pass through here, and this transport is going to turn the base plate round. So this will generate force here. Now, not all bacteria have flagella, and just like the O antigen of LPS, the proteins that make up the filament have a structure that is variable, varies between bacteria even of the same species. So this is also used in identification. It's the H antigen. H is for heat, because the antigen is denatured if you heat it up. Okay, so it's protein, it'll be denatured. Whereas the O antigen and the K antigen are heat resistant. So if you have a strain of E. coli, H70157, this is the serotype of the flagellum, and this is the serotype of the capsule antigen. Flagellar synthesis. Okay, you have the subunits that are synthesized in the cytosol. They're transported up the inside of this structure, and then they are attached onto the outside. And so the flagellum, extrudes or elongates by adding extra subunits to the tip. This is like what? Yeah, type 3 secretion PD. It's the same kind of thing. So this is where the flagellum comes from. Okay, it's an adaptation of a pre-existing structure, which is this. Okay, once you've got this kind of thing, you use it to inject protein subunits or secrete them. It can be modified to produce the flagellum. All you need to add is, the rotor structure, something that is going to transport the protons and produce force. You don't need to make the whole thing from scratch. So that's how it works. Now, the problem for the bacterium is how to use flagellar rotation to move about in an organized way. So and this is, okay, so flagellum, its function is movement in a liquid medium. Okay, type 4 PD are for gliding motility over a surface. The flagellum is for moving around in a three-dimensional space in liquid. So if you or I were designing how to move about in three-dimensional space, you'd probably need 3 sets of controls. So one to do like this, one to go like that, and one to do this. Okay, so in aviation, you call them pitch, yaw, and roll. So you need these 3 sets of controls. Here you've only got 1 set of controls. Here you've got the flagellar rotor, and it can go one way or it can go the other way. That's it. And you've got to try and move around just with those controls. So the way it works is like this. If the flagellum is turning around in an anti-clockwise direction, then the cell is going to move forward in a straight line. Okay, you can stop. Okay, you can stop moving as well. On, off, clockwise, anti-clockwise, and stop. So I guess you have 3 options. Now, if the flagellum starts to rotate in a clockwise sense, then what will happen is the bacterium is going to start to move around in a random direction. And that's true if you are monotricious or peritricious. The same principle applies. So how does this work to get bacteria to move in a way that kind of makes sense? So imagine you are a bacterium and you want to go and eat some glucose. It's here. It's diffusing out in the liquid medium from some kind of source. So you have a concentration gradient of the glucose. It's high here, and it's going to be lower the further away you get from the source. Your bacterial cell starts out around about here. And flagellar, so it's going to detect the presence of glucose and start turning its flagellar rotor in an anti-clockwise sense. So it's going to start moving. Now, if it's going down the concentration gradient, moving away, what is going to happen is the flagellar rotor is going to change its sense of rotation. So this cell is going to start falling around, moving randomly, and at some point after it's going to switch rotation back to counterclockwise. Yeah, clockwise, sorry. Hey, this is going the wrong way round. Yeah, it's because this one is wrong. Look, okay, that's right. Okay, counterclockwise. It's going to go straight. So it's going to start moving up the concentration gradient. As long as it moves up the concentration gradient, it's going to carry on, same flagellar rotation, and it's going to keep on moving. But of course, it's going to overshoot, right? It's going to go all the way past. And once it's going down the concentration gradient, then the sense of rotation is going to change again, and it's just going to start moving randomly around here. So the net result is that just by switching between these 2 senses of rotation, the bacterium is going to end up moving randomly very close to the peak of concentration. So chemotaxis is going to work in a logical way. Okay, that's enough for me. So I'd like you all to think about these questions. Which structures are useful for identification? While I'm doing this, I'm going to give myself an alarm for this afternoon so I don't forget, because I already stood you up once. Okay, for identification. Core oligosaccharide. Who says yes? Who says no? Everybody. Oh, repeats. Yes. See you, Pili. Of course, in Glasgow we say see you, Jimmy, but that's another. No? Okay, capsule antigens. Yes. Peptidoglycan. One person put their hand up. No. No. Okay. So what is useful for identification are capsule antigens, LPS repeats, and flagellar antigens. So why not this one? Why don't we use peptidoglycan or the core oligosaccharide for identification? They don't change. They're invariant. They're the same, always the same. So it's useless. What about this one? Why don't we use this? Not so. Wait. CUPD is just for adhesion. No, because it might use a different type. It might use a curly, or it might use the type 4. Yeah, but yes, but I think the answer is why aren't Fimbriae or PD used for identification is because they're too variable. Okay, so one bacterium will have several different types, and another strain, some of them will be shared, but some of them will be different. So the whole thing is too complicated. Whereas for the O repeats, on one bacterial cell, you've got one type. Okay, and all gram negatives are going to have these. So you have one type per cell. Same for the capsule. You have one type of antigen per cell. And for the flagellum, it's the same kind of thing, one or sometimes two. But for all of the Fimbriae and PD, you might have several different types per cell. So the whole thing is just way too complicated. So this is too variable. This is too conserved. Okay. Okay, just a few things to finish up on structures. So now we're going to the inside of the bacterium, which I have to say is rather less interesting than the outside. Okay, so what can you find on the inside of a bacterium? Inclusion bodies. So these are just structures for storing nutrients. It can be organic, glycogen, poly-beta hydroxybutyrate, so some kind of like, again, organic carbon. Cyanophycin, so it's a polymer of arginine and aspartic acid. So it's storing carbon and nitrogen. And some bacteria can have inclusion bodies for inorganic nutrients. So polyphosphate, phosphate can be limiting nutrient for many bacteria and plants. So if it can be stored in a granule, that's useful. Or purple sulfur bacteria store up sulfur when they have a lot of it. So that's what you can see here, these inclusion granules of sulfur. So these inclusion bodies, either they're directly in contact with the bacterial cytosol, or they can have some kind of protein or lipoprotein cage around them. But it's not really, complex membrane structure. So storage function. Some of them you can see on light microscopy, otherwise by electron microscopy. So this one here is about the only one you can really identify. There's a hole here in electron microscopy. That's because the poly-beta hydroxybutyrate is entirely dissolved by the solvents used in the sample preparation. It's very hydrophobic, so it's going to dissolve in the solvents. So you get a hole in the structure. That's a poly-beta hydroxybutyrate granule. What else do you have inside the bacteria? The nucleoid. Bacteria don't have a nucleus, but the DNA is compacted in some kind of structure inside the bacterium, because if you very, very gently digest all the protein in a bacterium, you can see here is the cell, and here is the non-compacted DNA that is spilling out of the cell. So in order to get all of the genome inside this volume, then the DNA can't be in a long extended filamentous form. It's got to be compacted. Now in eukaryotic cells, this is the histones, function of histones, but in bacteria, condensation is a function of polyamines like spermidine, and some nucleoid proteins. For example, there's one called the HU dimer. So they have the same kind of function as histones, but they have no structural homology, right? HU proteins. I don't know what that stands for. HU protein. So it has the same function as a histone. The exceptions are some groups of archaea have histones. And there are some weird bacteria, like this one, Gemata obscura globus. This is a really weird thing, is you see that the bacterial DNA is sequestered inside a membrane structure here. And there's even something that looks a little bit like a nuclear pore. So there are some weird things in the bacterial world that don't really conform to this general rule. And the last thing to talk about for general bacterial structures is the bacterial cytoskeleton. So as I said yesterday, classically the idea was that bacteria do not have any structure in the cytosol. And it's true that when people looked by sequence homology between the proteins of the cytoskeleton in eukaryotic cells, actin and tubulin, there were no homologous sequences in bacterial genes. However, there are proteins that have structural homology and perform similar functions. So the bacterial structural homologue of actin is MREB, that's in gram negatives, and MBL, which is for MREB-like in gram positives. So this is a protein that can exist as a monomer, and it can polymerize into filaments, like actin. And when you look inside bacterial cells, and this is with a GFP-tagged MBL protein in Bacillus subtilis, you can see that it forms these spiral filaments that are along the length of the cell. So what is the function of MREB-MBL? It's what pulls the bacterium out into a rod-shaped form. So of course, cocci, they do not have these proteins. They're spherical. So presence or absence of MREB, MBL, and structure of the cell wall, these are the two things that determine what you see in microscopy in the gram stain. The basic morphology of the cell, bacilli or cocci, and staining gram positive or gram negative. Now, tubulin also has a structural homologue in bacteria, which is called FTSZ. And that has a very, very interesting function in bacterial cell division. So, like actin, like tubulin, FTSZ can exist in a monomeric form or a polymerized form. During DNA replication, FTSZ will start to polymerize and form a ring, which is going to separate the two copies of the bacterial chromosome. And the position of this ring of FTSZ determines where the bacterial cell is going to divide. So the FTSZ is not itself, constricting like this and pulling and separating the two cells, but it seems to be the platform that determines where all the proteins are going to accumulate that actually do the job of separating the two bacterial cells. So this is very interesting, at least for me anyway, because what is one of the functions of tubulin? Gamma tubulin in eukaryotic cells has a primordial role in starting mitosis because it's very important for initiating the formation of the mitotic spindle. This is a tubulin structure. So you've got two structural homologs, very, very distantly related in bacteria and eukaryotes. These tubulin homologs, important in cell division and chromosome segregation. So probably, you know, the origins of the process of mitosis probably had a simpler precursor in some kind of prokaryotic cells using the same types of proteins. Even though there's like huge differences between bacterial chromosome segregation and mitosis, I don't know, it's really interesting that, you know, at some level the same types of proteins are being used for these functions. Okay, so the last bacterial structure you will need to know about are endospores, which are only formed by some gram-positive rods, gram-positive bacilli. You don't find endospores in gram-negatives at all. And these are in here because sometimes these are structures that can be seen by light microscopy. And the position of the endospore with respect to the rest of the cell is an element of identification. Whether bacilli, gram-positive bacilli are spore-forming or non-spore-forming, if they form spores, what do they look like? So, for example, central spore, that's very typical of bacillus. Subterminal, this is Clostridium. This thing here, you know, terminal or subterminal, I don't think you can really tell the difference here. Basically, if it's towards one end, Clostridium species. I don't know if you could really tell the difference between these two. So what are spores for? Just like for any other type of organism, they are resistant, inactive cells that are there just to keep the bacterium alive for prolonged periods and allow growth later on when conditions are favorable. Bacterial endospores are really, really tough. Okay, so they don't care if it's totally dry. They have no water, it doesn't matter, they survive. They are very heat resistant. You can heat them up to 100 degrees, boil them, and you can still recover vegetative bacteria afterwards, so they aren't inactivated by boiling. However, if you increase the pressure and heat to 120 degrees for 20 minutes, then you can destroy endospores. So that's why to sterilize bacterial medium, you don't just boil them up, you have to heat under pressure for 20 minutes at 120 degrees. Otherwise, your media will, you know, be contaminated, potentially be contaminated by bacteria that grow up from spores. So what we want to understand is what are they made of and what makes them so tough, right? So going from the outside to the inside of an endospore, the exosporium, sometimes it's not present, it's like a protein. Then you have two very thick layers of protein which make up the spore coat. So this is just giving you a big, thick, impermeable layer that is going to protect the bacterium from all types of harmful chemicals. Bleach, reactive oxygen species, things that can damage DNA. So you have a big protein barrier. On the inside, you have a cortex, which is a thick layer of peptidoglycan, just like the cell wall. But in addition to that, it's got this small molecule called dipicolinic acid, complex with calcium. And for a long time, this was thought to be necessary for the heat resistance. But in fact, mutants have been obtained that do not express dipicolinic acid, and apparently they are still heat resistant. And then on the inside, you've got a kind of dehydrated, metabolically inactive bacterial cell. So it's got the DNA, okay, the genome is there. You've got a cytosol with ribosomes, DNA repair enzymes, and SASPs, which are small acid-soluble proteins. So these are other proteins for condensing DNA. Acid-soluble, that means the protein is basic. So it's highly basic protein. It's condensing the DNA, keeping it in an inactive state. So at some point or another, this spore has got to reactivate. And this occurs when the spore will find itself in favorable conditions. Germination is triggered by the presence of nutrients, you know, like amino acids, sugars sometimes. And for some types of endospores, this germination has got to be preceded by heating. So you've got to have a heating step to activate them, and then the spore needs to be in the presence of nutrients, and then it can reactivate. So what happens first? What appears to happen is that the presence of nutrients has got to be detected somehow in the cortex. And this will lead to release of the dipicolinic acid, which is going to diffuse out from the cortex into the spore coat. Then it was going to activate at least one enzyme that is present in the spore coat. So this enzyme becomes active and starts to degrade the coat. So this spore coat will then be destroyed like this, and then this will allow rehydration of this bacterial cell. During the time that the endospore has been dormant, the DNA might have been damaged by radiation, so these DNA repair enzymes will very quickly repair all the damage. Rehydration will, start up the metabolism of the endospore, and then it will grow up into a vegetative cell. So that's germination and how are endospores formed, because before we saw that it's going to be difficult for a bacterium to synthesize complex structures outside the cell. Here you've got complex structures outside the cell, and the cell itself is entirely inactive in what's in the middle. So how can this metabolically inactive endospore synthesize anything? Well, it can't really. So the way endospores are formed is like this. So during vegetative growth, okay, bacterial cells divide, the two daughter cells are identical, and they both go on to grow and divide. During endospore formation, okay, got to be a trigger for this process, right? Either it's lack of nutrients. This is very often in the lab. What you can do is plate bacteria out on a very, very poor medium, so there's no nutrients around, and they'll produce endospores. It's not the only trigger for anthrax bacillus. It's the presence of oxygen, which induces sporulation. So if you've got an infected cow or a sheep, it will die. It's going to start vomiting, bleeding. All the anthrax bacilli that are in the blood, suddenly they're in contact with the air. And they realize, my host is dead. I've left my host. So they've got to produce spores to persist. So it's the presence of oxygen will induce sporulation. Okay, so what happens? So somehow the bacterium detects that the conditions are not good for continued growth. Lack of nutrients, presence of oxygen, something like that. First thing that's going to happen is the next division is going to be asymmetric. Instead of producing two identical daughter cells, one will be larger, one will be smaller. The larger one engulfs the smaller one. The smaller one then becomes metabolically inactive, it shuts its metabolism down, produces these small acid-soluble proteins, and just stops doing anything. And then it's the larger cell which will start secreting the cortex and then the layers of the spore coat. And in the final step, the larger cell will die and destroy itself and release the endospore. So here you've got two things happening in bacteria that are very unusual, perhaps unexpected. The first is differentiation of two cell types. During this process, okay, you've got bacteria, when they grow normally, each daughter cell is identical, they're all identical. They're carbon copies of each other. In this case, no, you get one mother cell and it produces two very different differentiated cell types. The endospore and the kind of like surrounding supporting cell. Differentiation into two distinct developmental pathways. Second thing that you have, programmed cell death of the bigger cell. It dies, as part of this developmental program. So these processes are absolutely essential for the development of complex organisms. Okay? In embryonic development, we start out from one single cell. To produce a complex organism, you need to have these two things, differentiation and programmed cell death. So they're already present in a very, very basic rudimentary form in some types of bacteria, these two fundamental processes. Okay, so I'm going to ask you to think about food for a little bit. So I'd like you to read this. And think about what is the best explanation. Everybody got it? Yeah, okay. So who says this? Okay, they're going to be killed during the first heating cycle. Nobody. Who says second one? Each heating cycle kills a proportion, so after you've done it 3 times, you get down to 0. Nobody. Who says the third one? They're activated by the first cycle and then killed during the second and the third. 1, 2. Okay, about half a dozen people. And who says, okay, they survived the whole thing? About 50-50. Okay, so the two answers that you've got here, this one and that one. The correct answer is this one. Okay, so they are in the presence of a very favorable nutritional environment. Capsule is a very rich medium, not just for bacteria, but also for anybody, I guess. So if they're heated, they're going to be activated, then they're in the presence of plenty of amino acid sugars, so they're going to start to germinate. They become vegetative cells. But if they are vegetative cells, they're not heat resistant anymore. So the second and the third cycle will kill all their activated bacteria. So that's what's happening here, this one.
If they become vegetative cells, right?
Yeah.
They won't produce more spores, or will they?
They won't, because they're still in a very favorable growth environment. If you took them out after the first cycle and then put them in medium with no sugar, no amino acids, nothing, then they would form spores.
Okay.
But you catch them by surprise. They're growing in this nice rich medium and then you heat them. No, I know, cut down in their prime. They won't have time to start producing spores.
The question is, why would heating it the first time matter?
Okay. So when I talked about germination of endospores, some types of bacteria require heating in order to activate them, and then in the presence of favorable growth conditions, germination will occur. So that was the link with this. And so this used to be used. This was a process called tyndallization, but now it's been replaced by heating at higher temperature, I think. Okay, so we have about 10, maybe 15 minutes. So I'll start on identification and taxonomy, and that way next week we can gain a little bit of time for metabolism. So in bacterial taxonomy and identification, there's always been kind of, and there still is to a certain extent, two approaches that coexist and kind of try and get fused together. One is identifying bacteria by their phenotypic properties, so phonetic classification and identification. So this is on a very practical level. I've isolated a bacterium from somewhere. I want to know what it is. And then, as well as that, so you've got to think about what it looks like, biochemical properties, this sort of thing. And then from this list of properties, try and construct a phylogeny. And this has been kind of overtaken by molecular phylogeny. That is the idea, okay, we'll sequence a load of genes and then try and reconstruct our phylogeny from the molecular data. You don't need to bother about performing a bunch of complicated biochemical tests. So these two approaches kind of coexist, but really in terms of systematics, it's molecular phylogeny that really is used now. So just like for the rest of biology, there's an attempt to try and put different groups of bacteria into this Linnaean style scheme where you've got 1, 2, 3, 4, 5, 6, 7 levels of classification from species to the kind of kingdom level. So this is the accepted standard system. So, for each bacterial species, you'll have a species name, a genus, and then family, order, okay? ACI, everything that's ace, right, is a family. And then ale, like that, that's the order. I go to a bar and I order an ale. And then these higher level kind of groups, class and phylum. Perhaps, well, it's always difficult to define what they, why it's a class and not a phylum. But anyway, so all bacteria are fitted into this classification scheme. So for identification, what are the kind of phenotypic characteristics that you can use? Well, the first thing you'll see if you've got some bacteria on a plate is you'll perform a gram stain and look at them under the microscope. So you'll have gram-positive, gram-negative, basic shape of the cell. If you use specific staining techniques, you can detect the presence of flagella. Look at the colony morphology, see whether they form spores. So these are a bunch of basic morphological characteristics, but this is not going to tell you a lot, okay? So you've got 1, 2, 3, 4 characteristics, so you can have like 8 categories with this. Colony morphology, you might have multiply that by 5 or something. So you could maybe distinguish maybe 40 or 50 different types of bacteria, just like this. But that's not really enough. Okay, so you need more phenotypic characteristics to be able to distinguish many different types of bacteria. So mostly this is reliant on bacterial physiology or metabolism. So their general nutritional type. What does that mean? Heterotrophic, autotrophic. Do they need organic carbon or not? Are they photosynthetic? If they require organic carbon, what can they use? What types of sugars can they survive just with I don't know, citric acid, something like that. Oxygen relationships, aerobic, anaerobic, facultative anaerobe, strict anaerobe. Mechanisms of energy conversion, what this is, it's photosynthetic, non-photosynthetic, whether they like, whether they're thermophilic, mesophilic, psychophilic, whether they like acid or not. So a bunch of growth characteristics here. And then for pathogens, whether your species is a pathogen, what kind of symptoms does it provoke? Is it a commensal bacterium? What kind of species can it infect? So something to do with interactions with other organisms. So you can describe a bacterial species with a kind of bringing together these different characteristics. So if you have these things, then you'll get an identification strategy, which can be summarized in this kind of identification key. So you'll get a colony. The first thing you do is perform a gram stain and determine whether you've got gram positives or gram negatives. Bacilli or cocci, if they're bacilli, do they form spores or not? Do they have a catalase or not? Cocci, catalase positive or negative. So you've got, morphology, microscopic morphology, some kind of simple biochemical test that you can perform just on the colonies on the agar plate. And this will give you, some kind of ideas about the identification to a genus level. So if you've got gram-positive cocci, catalase positive, it's going to be Staphylococcus. Catalase negative, Streptococcus or Enterococcus. If you have gram-negative bacilli, which are oxidase negative, then you'll have probably Enterobacteria. But there are a lot, okay? Escherichia, Salmonella, Proteus, Shigella, this kind of stuff. So you don't have an identification, but you've got an indication what they might be. And then once you've got that, so these are called orientation tests, then you can perform a whole bunch of biochemical assays to determine whether I've got Pseudomonas or Salmonella or Pastorella or Proteus, because these different types of gram-negative bacilli, they're all going to look pretty much the same in terms of microscopic morphology. So you need a panel of biochemical tests to determine which is which. So for each one of these tests, you'll score them positive or negative, and that will give you the identification. Okay, one more thing, and then I will stop there. Yeah, that's about enough time for that. So this is what you're going to do in the lab classes. You have an unknown bacterium, and you have to perform the orientation test, and then an API gallery, which is a set of biochemical tests. Now, in clinical bacteriology, this is being replaced by mass spectrometry. Now, how does that work? Well, you start off always by a colony on an agar plate. You prepare an extract and then spot it onto a metallic MALDI plate. So you can put, I don't know, an array might have 60 different spots, 60 samples on a plate. So the laser is going to hit the spot and vaporize all proteins and everything that's in the extract. They will be ionized and then go through the mass spec detector and give you a spectrum, which you can kind of see here. So you've got the mass charge ratio here, and then the peaks are, how many particles have that mass charge ratio. And then each bacterial species has a particular peak profile. So the companies that have developed this system, they've got a big database of the different bacterial species that they've tested by MALDI-TOF. So they know when they get a particular profile that corresponds to, I don't know, Enterobacterium fecalis, and another one will be Salmonella and this kind of thing. So you get the peak, and then the software just tells you what your bacterium is. And the reason this is very good is because you can identify a sample in 30 to 40 minutes, and you can do maybe 50 to 200 in a day. Now, if you're using kind of classical techniques like this, you've got the colony, you perform your initial tests, and then that gives you the more sophisticated tests that you need to perform. So this is day 1. Then you set up the test. This is day 2. So it's another day before you get an identification. So with the MALDI-TOF, you get the information on the same day, the identification. And this can be very important for clinicians because it might determine what antibiotic you need to prescribe to the patient. And if you wait 1 day to have this information, things can be a lot worse for the person who needs to have their antibiotics. So that's why, you know, the clinicians probably they'll start their antibiotherapy before they get the identification back, because they think, okay, this clinical panel, this clinical presentation probably is like this, so we'll give them whatever. Because if they wait for the identification, then you'll be losing 12 to 24 hours, maybe 48 hours, and things can degenerate quite a lot in that time. Okay, so I'll stop there with MALDI-TOF identification. So this is used in the clinical microbiology lab in Nantes. It's the routine now. But it's going to be a long time before we can get a MALDI-TOF machine in the practical classes here. So we're going to be sticking with the classical microbiological approach in the lab. Okay, so thank you very much. So I'll send out the question to you 4 p.m. If you don't see anything, send me email.